Hello, my name is Laura Duque and I'm presenting to you today the hepatitis delta virus histone mimicry drives the recruitment of chromatin remodeler for viral RNA replication. Okay, so first of all, I'm going to talk about the hepatitis delta virus, which is a satellite of the hepatitis B, which has a single stranded circular RNA, which genome only codes for one protein, the HDB antigen, devoid of any other RNA polymerase activity, and it needs the hepatitis D virus proteins for its own assembly. So, this virus causes hepatitis D, which is an infection that causes the liver to be inflamed, and this swelling can impair the liver functions and cause long-term liver problems, which can be the liver scarring and cancer, and it can only infect people which are already infected by the hepatitis B. Okay, so the virus has a single-stranded RNA genome and an enzyme called the reverse transcriptase. The one is in charge of copying the RNA molecule into a complementary DNA molecule, so that then once it is on the host cell, it can begin its own transcription, its own assembly. Okay, so in this case, the protein produced by the hepatitis delta virus copies the sequence located in the podocytes of the histone tree, so that then this sequence can be recognized by a broader domain called the BAC2B, that then it can begin the HDB replication and transcription using a polymerase II, so that it can invade the host chromatin, in this case the podocyte. The general objective of the investigation was to observe how hepatitis delta virus histone mimicry drives the recruitment of chromatin remodelers for viral RNA replication. So the first method they used was immunofluorescence, which basically they use antibodies conjugated to fluorescent dyes to evaluate cells in suspension, culture cells tissue, or for the detection of proteins. Okay, so in this experiment, they localized the wild-type SHDAG proteins in the nucleus using the podocyte cells, using an antibody for the protein, which was the hepatitis delta antigen, and the nuclei was stained using DAPI. Okay, so the PCR is an in vitro enzymatic reaction that amplifies millions of times a specific DNA sequence, or indirectly through the RNA. In this case, they use it to identify which isoform of this molecule are or is expressed in the podocytes. So then they used the Western blot, which is an analytical technique used to detect a specific protein based on its molecular height through the use of antibodies. So in the experiment, they used as an analysis of cellular fractions to see if this other form of the proteins, such as the wild type small hepatitis delta antigen, accumulates in the nuclear fractions. And the last one was the Northern blot. So they used it to basically identification of a specific RNA sequences that does not require specific or restrictive enzymes due to the RNA like small molecules. So in the investigation, they use it to analyze the effects of this other form of protein substituted in the small hepatitis delta antigen on the hepatitis delta virus RNA replication. Okay, so in the first results, you can see that the HDAG lane and the input lane shows and represents the 10% and the 12% of the volume of the whole experiment. This shows me that using these immunoglobulins, that are the proteins for each of the podocytes and the virus, you can see that in the affected cells, it shows a bigger precipitation of these proteins than in the non-infected cells. Also in here, but these ones are the little ones and these ones are the bigger ones, forms of the proteins in the podocytes. In part B, you can see that the PCR products demonstrate that the SNF2L and the SNF2L within the exon 13 are present in each of the cell lines, which are represented for all the podocytes and the hepatitis virus. On these results, you can see that they use 4 different eluates just to prove that using the 2 different immunoglobulins, the 4th eluate shows or presents the two different types of infected proteins that are the ones that are most concentrated than in the other forms of alloids that do present the histone 6 green fluorescent proteins. Okay, so in here you can see the immunofluorescence, which shows the two types of proteins, the wild type and the substitution one. And using the two techniques, you can see that inside the nucleus you have a similar pattern for both proteins. So you can see the green little dots that are both kind of in similar ways. In the second one, you can see that both of the same proteins are shown and expressed in the same way, in the cytoplasm and inside the nucleus, using the hepatitis delta antigens, the histone 3, which is the one that we're targeting so they can replicate, and the tubulin. On the third graphic, you can see the acetylation levels, which are kind of the same. As you can see, both have both condensed and concentrated proteins. Okay, so on the fourth one, you can see that when these cells are being treated with cyclohexamide, the one is in charge of inhibiting the protein synthesis, there is no change on the proteins produced by the virus. So that unique relation time, there is no change on the concentration of the proteins in the cells. On the last chart, you can see that the proteins that are being infected with some green fluorescent proteins, the ones are in charge of targeting the nucleus with a different form of a rhododomain just to inhibit these proteins, they don't see like any effect during the concentrations. But when they're being infected with a different kind of rhododomain, there is no concentration of the proteins. But they show that in the nucleus they have the green fluorescent proteins that are the ones that are being changed, and there is a major concentration of the histone 3, that is the one that the sequence original from the hepatocyte. So, on the last results, they use the human hepatocyte cells infected with the two types of the proteins from the virus. And you can see that during long and short exposition to the virus, the WT or the wild type small hepatitis delta antigens shows bigger concentrations during the short and the long incubation time. Okay, so in here you can see that also there is a major concentrations of the WT protein during the RNA replication than using the substitution protein. Okay, so in the discussion we used three authors just to compare that if they agree or not with the whole point of the article. So the first one said that the acetylation of the small hepatitis delta antigens protein plays a crucial role in the synthesis of the hepatitis delta virus RNA by cellular polymerase II. So that is completely right, because the whole point of the exposition was that these proteins are the ones important for the mimicry of the histone 3, and if they get bind to the rhododomain, they can activate the polymerase II, so they are right. The second author said that the histone mimicry is used by certain viruses to subvert normal host defenses. So this is right, because it was the whole point of the experiment, which the viruses attack other hosts just to replicate and have effects. During the third one, they said that the protein acetylation motif, so this is the sequence, mimics the histone sequence, is read by the rhododomain of the same rhododomain protein, chromatin remodeling complexes 18. So this is right, because the rhododomain actually recognizes the sequence given by this protein. It's right. Okay, to conclude, the HDB uses a mimicry to the histone 3 located inside the human hepatocytes, just to guarantee the RNA replication and to assure the persistence of the HDB infection, using an active polymerase II. The second one is that the proteins in the rhododomain enhances the HDB replication, and that the rhododomain interacts with the protein promoting the disease. The one was reassured during the whole experiment by the methods and the inoculation of the healthy hepatocytes with the mutant genes for the HDB. showing the interaction of the broader domain within the cell's original histone strand.